Hey y'all, here is your lesson on cell membranes, and also we're going to talk about transport, so getting stuff into the cell and getting stuff out of the cell that it doesn't need anymore. Okay, so the cells need an inside and an outside, so we need something that will separate it from its environment and keep everything contained. But at the same time, the cells need stuff to get into it. It needs food, so sugars, proteins, fats. It's also going to need some gas like oxygen. It's going to need some water. But at the same time, we want to get rid of some of this stuff. So we'll have some waste. Ammonia is an example. Or what if the cell at the ribosome is producing proteins but needs to transport it elsewhere? Well, we need to get those out of the cell as well. So basically, we need to get materials into the cell that it needs, and then products or waste, we need to get that out of it. So how do you actually build a membrane? So we have a high content of water inside of the cell, but cells are usually found in watery environments themselves. So we need something that won't mix with the water, and if you're thinking what I'm thinking, oil doesn't mix with water. So we're going to use lipids. Lipids and fats are what makes the membrane because it doesn't mix with the water. And so here's what the lipid looks like that make up the membranes. It's actually a special kind of lipid called a phospholipid. This is what it looks like. So it's actually got a phosphate head up here and then a lipid tail. And there's something special about these two things. So the phosphate, that part of the molecule, it's actually attracted to water. I don't know if you remember this from before, but to be attracted to water, that word is hydrophilic. So it likes being around the water. The lipid side is actually hydrophobic because fats don't like water, so it's repelled by the water. So you've got this hydrophilic side that likes water and this hydrophobic side that doesn't like water. And so if you notice, this is what the membrane actually looks like. And so they arrange themselves with their hydrophilic side towards the water, inside the watery substance of the cell and the cytoplasm. Their hydrophobic tails are on the inside together. So it's the heads on the outside. And that's why it's called a phospholipid bilayer, because it's made of two layers of phospholipids. Okay, the cell membrane is also said to be semi-permeable. Not everything that is found in the environment of a cell, the cell doesn't want all of that stuff to come in. It just wants what it wants. So it kind of needs a bouncer, right? You don't want all of the trash getting in. You just want the good stuff. So it's going to allow some materials to get in, and so a cell that allows only some materials to get in, that's called semi-permeable. These are examples of all the things that it needs. But which ones can actually just get in? So some of these things, like fats and oils, can actually pass directly through. They don't need any help getting in. And if you think about it, it makes sense. The membrane itself is made up of fats and oils, lipids, phospholipids. So of course other lipids are going to be able to go straight through because they're mixing with molecules of its same kind. But what about everything else? What about the sugars and the waters that it needs? Well, those, you actually need a special door or a protein channel that is going to allow those substances to come into the cell or leave the cell. There are specific channels for each of these molecules. So there's a specific channel for sugar, for amino acids, which is for building proteins, and then for water. So those are protein channels. They are to let specific molecules in. Here's what the semi-permeable cell membrane actually looks like. So we've got our phospholipids in here. It's our bilipid membrane. It's got two layers of phospholipids. The hydrophilic heads are on the outside. And then if you notice those protein channels, so proteins are special and they're perfect for being channels because they're not hydrophobic or hydrophilic. I mean, technically they are hydrophilic, but they like both lipids and water. So they don't arrange themselves only on the outside like this. They embed themselves in the entire membrane so that it can cross the entire thing. So it's perfect to get it's a perfect channel to get one molecule from one side to the other. So they're doors. The proteins are acting like doors. They're moving their specific molecules. And if you notice, I'm about to talk about this next, but it's moving from a high concentration to a low concentration. And it just goes through the channel like this. Okay. Molecules aren't just going to move by themselves through these membranes. It has to be part of a concentration gradient. So you're going to see these everywhere from now on, high to low. So what that means is that if there's a high concentration of molecules on one side and there's a low concentration on the other, well, the ones on the high are going to go into where there's a low so that you get an equal amount of both on each side. And that is called diffusion. So that is the movement of molecules from a high concentration to a low concentration. Here's a good diagram that illustrates that. There's a whole bunch of molecules of dye on this side of the membrane and none on the other side. So they're going to move this way to get to the other side to where you get an equal amount, and so you have reached equilibrium. This is passive transport. So basically what that means is no energy is needed. It's going to happen all the time. Think about it like a slide. When you start at the top of a slide and go down, you don't need any energy for that. What you need the energy for is to actually climb the ladder. But when you go down, you don't. Here's another example of the diffusion. Say you actually sprayed like a perfume in your room, in the corner of one room. Well, if someone was right here versus right here, the one all the way in the corner, it would take them longer to smell whatever that perfume was. And then again, the same thing happens in cells. Okay, so that's simple diffusion. So here's an example. We've got a cell. We've got 1, 2, 3, 4, 5. We've got 8 molecules of fat on the outside of the cell. We have 6 molecules of fat on the inside of the cell. So which way is it going to go? Well, one of these fat molecules is going to go to the other side so that you have an equal amount of fat molecules on both sides. So the cell is now at equilibrium. Facilitated diffusion is moving from a high to low concentration. It's still diffusion, but you're actually going to go through a channel. So we're using those protein channels. So now things like sugar can still do the high to low thing, but they just need a little extra help. It's like having someone help, like carry them across the membrane. So here's the difference. Simple diffusion is moving directly through the membrane. Facilitated diffusion is using help. It's using a protein channel. For both of these, a similarity is that no energy is needed for these processes to help, for diffusion to help. So here's another example. Simple diffusion over here on the left. A lipid can go straight through the membrane. Water or sugar or amino acids have to go through one of those protein channels for facilitated diffusion. Active transport, on the other hand, is going to be when a cell actually has to go against its concentration gradient. So think of it as it's going uphill. To go uphill, you need a lot of energy, or going like swimming against a current. That's really hard. You need a lot of energy. So for active transport, it's opposite of diffusion. You're actually going from a low to a high concentration gradient, and you have to use energy. And if just as a quick refresher, the type of energy we use in cells is called ATP. Protein pumps are going to be what helps accomplish this. You need a protein pump to pump these molecules against the concentration gradient. So here is everything wrapped up. Simple diffusion and facilitated diffusion are both examples of passive transport. It does not require energy. The difference between them is facilitated diffusion. You need a protein channel to help the molecules get from its high concentration to a low concentration. Active transport uses energy because it's a protein pump is pushing these low concentration molecules against the gradient. It's going backwards of diffusion, basically. Okay, osmosis, the definition of osmosis is that it is movement of water across a cell membrane. So water is really important, and so we have to talk about just water moving across the cell membrane. It's still considered diffusion, but it's just a special type of diffusion where it's just the movement of water from a high concentration to a low concentration across a semi-permeable membrane. So say you had water here versus here. The water is actually going to move from the left side to the right side, because if you can see, there's more blue, so there's more water. So even though there's more molecules on the right-hand side, we're just looking at the water now, the movement of the water, because this is osmosis. And so now you can see the level has gone down, it's balanced itself out. If diffusion was going to happen here, then some of these molecules would go to the other side. Okay, it's really important to keep water in balance. Our cells surviving actually depends on this. And also, this could be a great example in case you ever want to buy freshwater fish, because you don't want to put it in salt water. So this is an example. We'll go through each of these of what happens when you put cells into freshwater or salt water. Okay. If you put a cell into fresh water, another example of that is hypotonic. So make sure you know that, because I think that's what I wrote on the paper. So if you put a cell into a hypotonic solution, so into fresh water, there's actually a higher concentration of water outside the cell than inside the cell. So water is going to go into the cell to balance it out, right? But at the same time, the cell's gaining water, it's going to burst if it gets too much water in the inside. An example of this is actually paramecium. The same thing will happen to them. So they have this thing called a contractile vacuole that actually pumps water out of the cell since so much water is getting into it. In plant cells, they won't burst because they have that thick cell wall around it, and so their vacuole will fill up with water, but we want it turgid like this because of turgid pressure. And that's when stems are upright and your plants and flowers look really pretty standing up straight. Here's what the contractile vacuole looks like. So this is it all filled up, and then it decreases. Okay, what if you put a cell into salt water? Another word for that would be a hypertonic solution, which means you have put a cell into a concentration of water that's lower than inside the cell. So if you have a lower concentration of water outside of the cell, your water inside the cell is actually going to leave, and now your cell is going to shrivel. It's losing water. You don't want this to happen in plant cells, although they'll still be fine because their cell wall is rigid enough that it'll hold it up, but you still want to make sure, that's when you would see your plants wilting and you would want to water them. We want to keep a good balance of things, and this is called an isotonic solution. So this is when it's balanced conditions. So that means the rate of water going into the cell equals the rate of water leaving the cell. So there's no difference. Your animal cells are normal, but for your plant cells, this is just flaccid. This is just like meh. You really want your plant cells to be turgor in a hypotonic solution. You want them to be as full of water as you can because you're not going to break your cell wall. So this is when the cell is in equilibrium and the volume of the cell is not going to change. And I believe that's it. If I rushed through things, please come into tutoring or email me or rewind it and start over. Okay, see y'all tomorrow. Bye.